For coming, and today we have 3 lectures. First, I want to introduce Germán Rosano. I know Germán Rosano for how many years? I don't want to count how many years.
Too many to remember.
I was in the middle of my postdoc, and Germán Rosano entered it in the lab. And
Yes,
cool,
because I'm much younger than you.
Yes, slightly. And I could guarantee that he has the same highlight he has right now, so he has to be growing physically during this year, but he has made a fantastic career. Right now he's researcher of the Argentinian Council of Science and also is vice director of the platform of unit of mass spectrometry at the IBR. And in his lecture today he's going to introduce network analysis. Yesterday we talked a little bit about, or the speakers were presenting interaction between proteins, techniques and so on. And right now, Germán is going to put everything together and give us an idea about, could we really unveil networks like proteomics?
Obviously. That's it. Thank you, everybody. Hello, everyone. I hope you had a lovely evening last night after the cocktail. And as Nico said, I'm going to introduce to network analysis. This is the computational part after the experimental part of interactomics. And the thing is that after a proteomics experiment, we can reveal a lot more information than just a list of differentially expressed proteins. So we are going to talk about that, how we can use network analysis to reveal much more information than just a list of proteins. So, what is the importance of protein-protein interactions or PPIs? Well, as already all of you know, proteins have many functions. So one protein can participate in different pathways and different networks. So it is really important to understand that we have a multiplicity of functions and networks that are working together. So it is important to comprehend a protein's function and behavior in order to understand and predict biological pathways. And also, these functions in protein-protein interactions can lead to disease, or what we can do also is to target some interactions as a pharmaceutical option. And networks are everywhere. Networks are interconnected units that travel information from one part to another. And we have biological networks, we have social networks, and the underlying analysis of networks are the same just for a social network or biological network. And we're going to learn today about core analysis of these networks. So, as I told you in the beginning, yesterday and today we are going to learn a lot about the experimental part of interactomics, and in this lecture we are going to see about network representation and network analysis and visualization. So, first of all, a little history about network analysis. This began in the 18th century, more or less. There is a great mathematician called Leonhard Euler that at that time was interested in one question about a simple common problem, a real world problem, that is now famously known as the 7 bridges of Königsberg. Königsberg was a city in Russia that has a river going through it, and this river forms 2 different islands surrounded by the landmass, and there were at that time 7 bridges that connected the different masses. So Euler came up with a question saying, well, is there a pathway where I can go from one part to the other, crossing each bridge only once? I know everyone as a kid may have done this type of exercise, trying to join dots without lifting the pencil, something like that. And for example, we can start here, going here, then here, here, and then we can go here, but I already crossed these bridges. And again, we can start doing this exercise to see if we can make a path crossing each bridge once. So Euler was, at that time, concerned with that question. So what he did was to come up with a whole field, a new field in mathematics called graph theory, in which he answered this question and said, no, there is no way we can do this with this type of problem, because we have 4 points, four land masses connected by these bridges, and each point had an odd number of bridges coming out or coming in of it. And the mathematical proof was that if you have an odd number of links between points, you cannot go through all the points only crossing one point once. So, and also what he discovered is that it doesn't matter the relative position between the points. It is completely irrelevant. Only what we need is the number of points and the number of links going through each one of them. So, thanks to that question, a new field of mathematics was born that is called graph theory, and we are going to learn a lot of concepts about graph theory and how can we apply graph theory to biological networks. And he also, as I said, he discovered that position is completely irrelevant. We can move all the points and the conclusion will be the same. And in doing that, he laid the groundwork for topology, that is another field of mathematics and geometry. So, basically this is a network. We are going to see a lot of this graph today. And we can start naming things with the correct name. For example, in this network, what we have, an Euler called them vertices, or vertex in singular. So a vertex is a set of objects also called nodes that are connected together. In protein networks, we are going to call nodes, but in other fields they are called vertices. But for protein, they are nodes. And edges, the edges are the connections between the nodes, are also called links, but for protein interaction, again, they are called edges. And as I told you earlier, we have networks everywhere, so we can think of anything, and for example, we can have a network of airports or airline networks, and the vertices would be the airports, and the edges would be the routes connecting the different airports. But of course, we can have more information behind these different nodes and edges, and that is called an attribute. An attribute is any kind of description that we can make from this node or these edges. For example, in airports, we can have the number of hangar, the number of staff population, and whatever. It's new information that we can add to the node. And for edges, the same. If we are talking about routes, we can talk about the distance, the weather conditions. These are called attributes. And in protein interactions, also nodes and edges have attributes. The type of edge determines the type of network. And for example, here we have an undirected network in which the edges don't have a directionality. For example, here we know that maybe F interacts with G, and it doesn't matter the direction of the interaction. We just know that each interacts with each other. So this is an undirected network. Obviously, a directed network has a directionality, and for example, A interacts with B, and then B goes to interact with the other nodes. And a weighted network is one where the edges have some kind of attribute where we assign a certain importance to the interaction. For example, the interaction of node A with F is, for some reason, more important and for the network than the interaction that with A with B, for example. And this is a weighted network. You may realize that we see these types of networks all the time. And in biological networks, we have all the networks that we know of in natural science. For example, if these nodes are protein or metabolites, the molecular metabolism pathways are directed networks, but protein-protein interaction networks tend to be undirected. So in interactomics experiments, we are going to see undirected networks. Of course, cell signaling is a type of directed network. So we're going to start describing these networks in terms of the concept that we just saw. We can see that a node can be connected to the other node, and the number of connections, the number of edges that go from one node to the other, and the total summation of the edges is called a node degree. So we can see here that this network has, this node painted in black, has a degree of 8. This node here, painted in black, has a degree of 3. And for all networks, we can assign the node degree to its node. And this is important to calculate the nodes because we can visualize the network to reveal which nodes have the higher degree, because that can be important to understand our network. And keep in mind that loops are counted twice. So this node have degree of 5, 2, 3, etc. So, as I said before, we can then visualize our network in terms of node degree. And we can have a new layer of information besides differentially expression that is also degree. And this is important because when we do a proteomics experiment, when we have those lovely volcano plots and we see, oh, we have 100 proteins that are differentially expressed, and maybe the question is, which one are we going to keep studying, all right? And we tend to focus to the protein that is farther away from the volcano plot because it has the largest fold change. But that is only one part of the history. For example, maybe this protein here that only have a degree of 1, maybe the fold change is 4, is the largest fold change in our proteomics experiment, but in our network is really far away from the other nodes and only have a connection with one other protein. So why are we assuming that because we have a large fold change, that protein is important? It's not always the case. So network analysis is going to help us with that question. And we can reveal, for example, that this protein that has a node degree of 5 may be, I don't know, may be more important because it has more connection, but maybe its fold change may be less, I don't know, maybe 2. Keep in mind that node degree is a local type of centrality. Centrality is, we are going to see a lot of metrics called centrality that indicate how the different nodes are connected in the network. So degree centrality, which is only one of these metrics, is a local centrality because it's only keeping into account the interaction of one node with its neighbors, but it's not taking into account the whole network. So it is a local type of centrality. And later on we are going to see more about global centralities that can help us to understand better our network. Calculating degrees is easy when we have 10 different nodes connected by a few edges, but in larger networks, when we have thousands and thousands of nodes and thousands of edges, the software that can do these type of calculations all work the same with an adjacency matrix. An adjacency matrix is just a two-way matrix where we put all the nodes in the vertical axis and in the horizontal axis, and we start counting the different edges that connect the pair of nodes. For example, here A with B have one connection, so A with B have one edge, and A with C, one edge, and so on and so forth. And for A, the summation of the edges is the total degree. For dielectric networks, we have, of course, this part is a mirror image of this part, is completely the same. But for dielectric networks, this part of the adjacency matrix is the out degree, is the number of edges that go out of the node, and this part that is not calculated here in the figure are the in degrees, the degrees that are coming into the node. And for weighted networks is the summation of the parameters that can be anything measuring the interaction between the nodes. Is the summation of the different weighted degrees. Adjacency matrices are a very good visualization for finding clusters. For example, Here we have an adjacency matrix of co-occurrence of characters in a book. The book is Les Miserables. And each time one character was interacting with another, someone took the time to count how many times these two characters were interacting. And this person made an adjacency matrix of all the characters. And we don't have numbers, but we have different colors according to the amount of interaction. So here we can't see anything, but if we apply different algorithms to try to unveil different patterns for clustering, we are going to see clustering later on, we can detect really easily and visually different clusters of characters interacting together. Because in this adjacency matrix, the characters are ordered in alphabetical order. In alphabetical order, we don't see anything. But if we cluster this, we can start seeing patterns of interaction between the characters, and of course we can apply clustering to protein networks, and we're going to see that later in the talk. And of course we have clusters of characters interacting together, and we can reveal which is the character that has the most interaction in the book, because we see that, for example, this character has a lot of interaction across the adjacency matrix, and it's the main character of Les Miserables, that is Jean Valjean. So, again, degree centrality can help us to reveal proteins that have a lot of connections. And these proteins that have more links that exceed the average of degree are called hubs. Hubs are proteins that have a lot of connections. They can be important or not. We are going to see that in a minute. Another important metric is the shortest path. Shortest path is, like here in the definition, is the shortest distance between any two nodes. And that determines the network diameter. The network diameter is the largest, shortest path in the network. So for A to B, we have 1, 2, 3, 4, 5, a shortest path of 5. And I don't know if this is the largest path. Maybe you have here 1, 2, 3, 4, 5, 6. Maybe the network diameter for this network is 6. And shortest path is going to be an important metric for other centralities that we're going to see in a minute. Another concept in network topology is transitivity. Transitivity is the probability that a node has to have adjacent nodes really interconnected. Transitivity measures the interconnectivity of different nodes. So, by a transitivity study, we can reveal protein communities, proteins that are working together in a cluster, maybe revealing a protein complex. And transitivity calculations are somewhat easy, and for example, if we have here this network, you all know that if we have 3 points in a plane, that determines a triangle. The amount of triangles, the amount of possible triangles is a calculation in transitivity, and here, for example, we have that for this network, the amount of triangles that can be formed between those 5 nodes are 4 triangles, but in example B, we only have one full triangle and no diagonals. So for B, we have 1 triplet and 4 possible triplets. So the transitivity for that network is 0.25. In here, in case E, we have transitivity of 1 because we have the interaction among all nodes. So this may be a protein complex working together. So transitivity, also known as the clustering coefficient, is another metric that we're going to use to understand and reveal protein complexes. And again, when we are deciding what is the best way to visualize our network, we can choose to go with degrees, no degree, that we saw earlier, but we can also visualize our network according to clustering coefficients. And of course, in here, A has the largest node degree, but this network is visualized according to clustering coefficient, and it is very easy to see that here we have a protein, maybe a protein complex, here another, and A is not participating in a complex, even though it has the largest node degree. So it's another way to visualize our network. So now that we introduced two core concepts in network analysis, we're going to apply these concepts to specifically to protein-protein networks. And through the study of this number for many years of protein networks, there were a lot of concepts that arise that protein interaction has in common. For example, PPI are always clusterized. They are protein-protein networks form a lot of clusters, proteins working together. You all know that it's very difficult to find a protein that works alone. In general, proteins tend to interact with each other. This interaction can be physical, so two proteins directly and physically interacting together, or it may be functional, maybe two different proteins acting on the same substrate, for example, maybe molecular chaperone with a protease. They have a functional interaction, but they are not working physically together, maybe. Another thing is that protein networks has what is called the small world effect. We saw earlier that one of the metrics is the shortest path. So since protein networks are highly interconnected, the shortest path tend not to be large. And that is called the small world effect. The small world effect, we can make an example in social networks. Social networks are also small world effect networks. Because it is very probable that I can be connected to any person in the world through Facebook in only 6 or 5 or 7 contacts. Because I know someone from, I don't know, from Europe, and then someone knows someone from Asia, and then someone from Asia knows someone from Africa, and in 6 steps, I can be connected to any person in the world. It is very probable. And social networks are small world effect networks, and protein-protein networks are small world networks as well. So the protein networks have great connectivity, network diameter tend to be small. That ensures an efficient and quick flow of information between the proteins in the cell. But what happens is we delete one of the nodes. What happens when there is a perturbation to our network? Is a protein network resistant to perturbations? We're going to see another concept, that is that protein-protein networks are also escape-free. What is that escape-free network? In escape-free network, we have this kind of graph that represents a log function in which we have a lot of nodes that have a degree of 1 or 2, or a very low node degree, and we only have very few nodes that have a large degree. So that is escape-free network. And it means that these networks have a lot of nodes connected to each other, but they are all connected to hubs, and the hubs are the ones that connect the different networks together. So they are called scale-free because if we add or we delete one of the proteins, nothing happens, because if we delete this node, the rest of the network is completely stable. Of course, they are vulnerable to targeted attack, because if we target a hub, then yes, things may happen. For example, I don't know, P53. That is a very important oncogene, or transcription factors tend to be hubs. That is why protein networks are very stable, and we all have the case that we are working our protein of interest, and we make a knockout, and we don't have a phenotype. We always want to have a large phenotype, because a large phenotype will lead to a nice correlation, but you need to understand that protein-protein interactions and protein networks are scale-free, and in most cases, we don't have a large network perturbation if we delete one node. So in contrast, random networks, if we delete one node, we start losing a lot of the connectivity between the network, but that doesn't happen a lot in protein networks. For the protein I study, CLIP-S is a protein that has a beautiful function, selecting proteins for degradation, and when we started studying, we thought, well, if we delete this protein, a lot of things will happen, and in bacteria and plants, whenever, if you delete this protein, really nothing happens. We don't have a phenotype. So, how we are going to visualize our protein networks? Here we have a really lovely graphic of a small network, but in publications or in theses or in seminars, we have a lot of information, and we tend to show this kind of graphics. They look really cool here in the screen. Oh, we have a lot of data, and I can make this beautiful network. But really, we can extract a lot of information from here. And people in the field of network analysis, when they see this type of graphic, they call it a ridiculogram. So please resist the urge to show a network like this full of interaction, because you can extract really any information from it. So we are going to see now how are the best ways to visualize a protein network. But before we do that, we have to understand where the data come from, and we are going to make another steps that I'm going to introduce. So the workflow of PPI network analysis and visualization is something like this. Of course, the first thing is we have to obtain the data, and we have to obtain the network. Then, once we have the network, we can perform centrality analysis. Then we move on to topological clustering, and then we can do another options. So let's start with the PPI network. Where are we going to obtain the data? So the data can arise from a lot of experimental setups. For example, of course, from an interactomics experiment. I'm pretty sure that Professor Deckel is going to talk about interaction analysis in the experimental part. Rosario talked about yesterday about wire attenuation, proximity labeling, and I was introduced to cross-link mass spectrometry. And there you have a table. All you need is an Excel table saying, well, this is my bait, and it's interacting with these proteins. That's all we need for making a network. We can make it manually. In fact, in the afternoon, when we're going to do the exercises of network visualization, it's just a simple network created manually in Excel. All that we need for an undirected network is two columns, and saying, well, okay, this guy interacts with this guy. This is a network, for example, of Marvel heroes from the comics. So we know that, I don't know, Black Panther interacts with Princess Santa. So that's all we need. And from that, the software can pick up this information and create the network. For protein interactions, we also have databases that recollect the experimental work from different groups. For example, one of the most famous is Intact, where we can retrieve networks that some people have already studied. Because maybe first what we have to do is to see if we are going to make an interactomics experiment, we have to see if someone didn't make it before us. Also, we can have a network from a proteomics experiment that is not an interactomics experiment. For example, this is quite common, a proteomics experiment where we reveal differentially expressed proteins. We can use databases such as string to retrieve interaction between these differentially expressed proteins, the upregulated or the downregulated. We can query the string database with these proteins and to retrieve information from the literature if different proteins are interacting together. And this is quite important to to know. We're going to make an exercise today in the afternoon, because sometimes I see that when people receive the volcano plots, they start Googling each protein to say, oh, I found a paper that this protein interacts with this one, but we can have this in string, and we don't have to spend 2 months reading a lot of paper, because string can do it in 2 minutes. So, when we have our tables, that is very easy to make. Of course, not the experiment, the table. But we move on then to centrality analysis, and we are going to see also the software that we're going to use for that. But first, we have to understand another type of centralities that are going to be useful to understand our network and to visualize our network. I already introduced to degree centrality, no degree centrality, and we talked about that is a local type of centrality. For example, this node has a relatively large node degree, but it's really far away from the rest of the network. So it may have a really large degree, but if it is outside of the whole network, it may not be that important. So, and we also saw that degree centrality is a really good metric to find hubs. So we need to move on to more global type of centralities to help understand the network. And one of those centralities is closeness centrality. Closeness centrality is a way of detecting nodes that can spread information very efficiently. So we're going I'm not going to talk about the formula, because we can do it here in an example, and we will understand easier making an example. So, for example, if we have here a really small network, we can make another matrix, but we are not counting degrees. This time we are going to count the shortest path of one node to the other. For example, from A to B, they are interconnected, so the shortest path is 1. From A to C, the shortest path is C. But what about A to E? We can go through here, 1, 2, 3, that would be a path, but the shortest path is 2, because A, B, and E. So the shortest path from A to E is 2. So we count each shortest path from each node to the others, and the summation of the shortest path is called the farness. Farness gives you an idea of how far this node is from the other nodes to the rest of the network. And the closeness centrality is this division, the number of nodes minus 1, divided by the farness. And the higher the closeness centrality, the closest is the node to the rest of the nodes in the network. For example, we can see here that B is a node with a closeness centrality of 1. It means that this node is very close to the other, and the information can reach easily from B to the other nodes. In contrast, D and E are the farthest away nodes from the network. So as you can see, since we have here the total number of nodes and the shortest path to all the network, this is a type of global centrality. It's take the whole network into account. So this again, high closeness score have the shortest distances to all other nodes. And here we have another visualization of a network in which we are painting the nodes with highest closeness centrality. Again, this node has the highest node degree, but this too has the highest closeness centrality, because if we need to reach from this part to this part, these two are close to the two sides of the network. So that's another type of centrality that we can analyze. Another one is betweenness centrality. And betweenness centrality is a measurement of the flow of information of a node in a network. It gives you that information, how a node is needed to pass information through the different through the different parts of the network. I'm not going to talk about the mathematical formula to calculate betweenness centrality, but it is only needed to say that the betweenness centrality detects the amount of influence a node has over the flow of information in a graph. Because here it is easy to see that this node has the highest between the centrality, because if we need to spread information from here to here, we have to pass through this node. There is no other way. So between the centrality give you an idea of how many shortest paths go through that node. So this type of nodes are called connector hubs, bottlenecks, bridges. They are interchangeable. And they are really important. You can see here that this node only has a degree of 2, but if we delete that node, the whole flow of information of the network becomes destabilized. So it is also important to detect this type of nodes with betweenness centrality. So basically, we have a lot of metrics, and we have to decide which is going to be the most important for our network visualization. So to sum up, here we have a node with a high node degree that is has a lot of connections, but here we have a node that has a high betweenness centrality because it connects the 3 different clusters, and B, for example, is really close to all of the other nodes. So there are different ways to understand our network. And I'm not going to talk about PageRank because PageRank is applied to directed networks, but I wanted to show because this is how Google ranks the pages in the internet with PageRank, and it's a measurement of how the different nodes are connected together and how the flow information goes to one node. So if we have a page that other pages linked to it, that ranks higher in Google. Well, this was the first algorithm. Now it's refined, and just a colorful comment that PageRank page that has nothing to do with web pages. Page is because of the last name of Larry Page, one of the founders of Google. So again, we can paint our network according to the different centralities, and we are going to reveal different information. In here, this network is colored according to degree centrality, node degree, and we have nodes that are highly connected. This is painted according to closeness centrality. These are the nodes that are very close to one another. And this one is painted according to betweenness centrality. And we detect nodes that are very important to connect the different parts of the network. So we have to decide which is better for us. This is painted according to PageRank. And again, the different metrics and how we are going to interact one another is different information that we can extract from our network. I'm not going to discuss this table. We're going to move on. So now, of course, that centrality analysis is all done by the software. We're going to see in the last few slides. And also topological clustering is going to be made by the software. And it is important to, when we see the full network full of nodes, if we can detect different clusters that are working together. That is called topological clustering. And it can detect different network communities, proteins or whatever object working together. Clustering analysis is also very important to maybe predict the function of unknown proteins, because if we do a network analysis and we find that, for example, this protein that is unknown is really close and in a community of all these proteins that we know is a common complex, we can apply one concept that is called guilty by association, and we can maybe say or predict that this protein is part of the protein complex. So if one protein enters in one cluster after topological clustering and we don't know which protein it is, well, we can say maybe that that protein is a new member of a protein complex. So, algorithms to make topological clustering are based on clustering coefficient that we saw earlier, transitivity, and a lot of other metrics like density, that is the ratio of the number of edges over the maximum number of possible edges. Conductance is another concept that is, if you see this community, we see that these nodes are hardly interconnected within the community, and we don't have those many edges coming out of the community. So that is a measurement called conductance. So clusters have many edges within it, and few edges coming out. Clustering algorithms, they are like, I don't know, 30, 40 different algorithms. They all rely on different formulas, and since clustering is an active area of research, that's why we have a lot of algorithms. But for protein interaction, there are 2 or 3 that are the most popular. For example, the Newman-Girvan fast-read algorithm or ENCODE. And today in the afternoon, we are going to use ENCODE and MCL. MCL is a type of algorithm based on Markov clustering that are very popular for protein interactions. And to understand really fast how one algorithm works, for example, the Newman-Girvan fast-greedy algorithm works something like this. If you have a network, the algorithm calculates the betweenness centrality of all the nodes and runs the betweenness centrality and start cutting each of the edges and recalculates the betweenness centrality. So, for example, if this network is here it starts with step 1, but there is a step 0, and with these 2 nodes are connected together, and you can see that this edge will have a really high betweenness because it is connecting the different parts of the network. So if we cut this edge, the edge betweenness is going to be completely different from the previous network. But if we cut this edge, the betweenness is going to be relatively the same from all the other for all the other edges. So the algorithm works like that. It starts cutting edges and recalculating betweenness between the edges. So later, when it does that for every edge until we have no edges left, and then with all that information, the algorithm starts clustering the nodes together. So finally, how are we going to visualize our network and with the hope to avoid making ridiculograms? So I have here a couple of examples of really cool looking networks from different papers. We can, for example, this is an interaction of HIV proteins with human proteins that interact on this. And we can paint differently the proteins that are coming from HIV, and we can make larger we can make the node larger according to the degree, and we can paint different things. Also, the border of each node can mean anything, for example, fold change in a proteomic experiment, and so on and so forth. We can also do gene ontology to our network and enrichment map to see if nodes that are clustered together also participate in the pathway or in some type of molecular function. So we can make this type of visualization. We can add images. This is, of course, not a protein interaction network, but it's really visually really cool. So, which software are the most popular to make network visualization? There are 2 or 3. Gephi is free open source desktop tool to do visualization, and the only thing that we have to do is to load our Excel table, and all the nodes are going to appear, and we are going to click on network visualization settings, and we can really easily, in a couple of minutes, we can have from a ridiculogram to a cool looking network like this. We also have Navigator, that stands for Network Analysis, Visualization and Graphing from the University of Toronto. And it's very similar to the others, but Navigator has also this type of visualizations where we can make these matrices according to the proteins and the different functions that they all have. And in the afternoon, we are going to work with one of the most popular, if not the most popular, that is Cytoscape. Cytoscape is, again, as Escapefree and Navigator, they are open source. The cool thing about Cytoscape is that they have a lot of plugins. So different computational researchers can code different plugins to see whatever, and we can upload this plugin to Cytoscape and start doing a lot of things. For example, today in the afternoon, and in the different day, we are going to use a couple of plugins to make this type of visualization. We are going to use Omics Visualizer and the String App, the Cluster Maker plugin to detect our cluster in the network. So finally, as this last slide, I'm right on time. The aim of this lecture was to introduce you to the core concepts in network analysis and to give you more tools to extract information that is maybe hidden in our proteomics experiment. Again, now that we have here our mass spectrometry unit and many of you are going to design your experiment, of course it is very nice if you have this type of volcano blocks. That means that our proteomics experiment is working really well. But from there on, we can use all this information, and please don't start focusing on this plotting because it's farther away in the fold change. We can make a lot of cool things to maybe detect the hubs, the bottlenecks, and maybe there are other proteins that may be borderline here, but maybe through network analysis they may be more important. And of course, another aim of the lecture is to say goodbye to this guy and avoid doing this type of visualization. Thank you.
Thank you, man, for the great talk. I want to say goodbye to this guy. This looks cool.
He's going to do it. Put it in your paper.
Do we have any question for Germán about networks?
Hello, Germán.
Hi.
Thank you for the presentation. I have been guilty of this notes a few times. And
Common in phylogenetic trees also.
I was wondering, a big part of what you're showing will depend on setting thresholds. Are there any set of guidelines or good practices for setting the thresholds? Are those published somewhere, or it's just like your exploratory analysis?
Yes, it's somewhat exploratory. The cool thing about Cytoscape is we're going to see this in the afternoon. When you are going to paint the different nodes in Cytoscape, you can use different mappings. Mappings mean that you are going to give different colors according to the centrality you are using. So if you apply a discrete mapping, yes, you are going to manually put thresholds that are going to, you are going to determine the threshold. So we don't have a guideline for that. But Cytoscape has another type of mapping that is called continuous mapping, and it works with gradients of colors. So what we're going to see today that if you want to paint all your network according to no degree, it is better to use continuous mapping and to use a color palette with different colors. I choose from a gradient, maybe from blue to red, and everything will fall within that threshold. And Cytoscape uses the range. For example, you will have degree maybe from 1 to 100. You can cut your range if you want, but you can use your full range to color all your nodes. But to sum up, there is no threshold. You are going to apply your coloring to your network according to what looks best for you.
Thank you.
Question?
If I you very much, Germán. Very nice. For me, a new world. I don't know if I lost it, but how do you, the program, calculate the length of the edge?
You mean?
The distance between
Oh,
nodes.
but in network, the distance of edges is irrelevant.
Okay.
So,
okay.
especially for protein networks, but in general for networks, position, o sea, the distance is not relevant. What you can have is an attribute to that edge. For example, maybe you know your constant of equilibrium of the interaction between two proteins, and you can make another table with the edge attribute, and you can use the thickness of the edge to represent different constants. But distance is, in fact, we are going to see in Cytoscape that I can go here with the mouse and grab this node and make it go away, but the network would be exactly the same.
Thank you.
Also, for example, one second, I wanted to make a comment to Jorgelina. For example, here, these are two types of representations. This is called a circular layout, and here, maybe in the beginning, all these proteins were in different places, and we can select this in Cytoscape and apply circular layout and get closer together. So distance is not important, but you can do all these type of things, and the distance, you will set the distance according to the nice figure you want to present in your paper.
I want to thank Germán for this wonderful talk. I'd like to, if you allow me, to make a contribution. Sometimes the connection between two proteins come from even an old paper and from a study that has been produced several years ago. So it's important when you work with this kind of schemes to learn perfectly about your protein. You have to search all that is known about your protein, and sometimes it's good to visit some kind of database like String that is from the Swiss Institute of Bioinformatics. There is the EMBL base for this kind of interactions. So that to search in the literature and to challenge your scheme with that information. Sometimes you have to clean, because proteins that are not in the same space shouldn't be contacting, and there may be a result that is coming from the lysis and the procedure. So it's really wonderful, and I don't think that there is this kind of huge. This only useful thing that makes you to think about the complexity of what kind of things you are confronting. You can say that, for example, if you compare a human cell, it's like the actual internet network. So this is the challenge you're facing. Something is like the whole network of internet is the network of more small molecules and proteins that you have in a human cell today. So this is really something that should be taken with caution, and it's really wonderful to have this kind of software to manipulate the data and to understand the data. Thanks.
Yes, of course. And regarding databases, what Eduardo said is very important. String captures information from papers and it's not manually curated. Intact is manually created. So Intact is really cool to retrieve good quality network, but in String you can retrieve whatever comes from the abstracts of paper. You have a slider where you can put the confidence. There is a one metric where you
Yeah,
can.
you have to put your criteria.
Exactly,
you have to go
exactly.
to the paper,
Yes.
read it, and say, well, that's rubbish,
Yeah.
it's good, and then you keep it or you throw it away.
Yeah, in string you can also say how many plugins do you want in your network. You can put, oh, I want to retrieve 200 interactors and they capture everything from. Exactly, yes.
Next question? Okay, we say thanks, Germán, for the
Thank you.